People are still coming in. Okay, whenever you're ready.
Great. Thank you very much for joining us tonight for the COVID vaccine Town Hall Trusted Messenger campaign. I'm Vishnu Chandi. I'm the chairperson for the COVID-19 task force. Joining me tonight is Dr. Edward Lin, excuse me, who is also part of the COVID-19 task force, as well as a trustee of the Chicago Medical Society and a professor emeritus in OBGYN from Rush University Medical Center. So today's been momentous because, as you saw, the new administration has a goal of vaccinating 100 million people in the first 100 days. A recent CNN vaccine acceptance seems to be increasing, predominantly interestingly, also splits on party lines, but also the Republican side is starting to increase their vaccine acceptance. Now it's about 2/3 of the US is willing to take the vaccine. And at the same time, the virus is also mutating and becoming more easily transmissible. And we expect the main virus, which is in England, to be the predominant strain throughout the United States. So can we get to the next slide? So Dr. Lin is going to talk about vaccinating pregnant and lactating patients against COVID-19. And then he's also going to initially start off by talking about how the vaccine actually works. So Dr. Lin, you're on.
Thank you very much, Dr. Chandi, and I want to thank the Chicago Medical Society for making this opportunity possible. May I have the next slide? So a question that I constantly get is, what is an mRNA vaccine? And I want to take a little moment to go through these illustrations with you to try to explain that this is a very novel, relatively new, but not completely new, because this technology has been in existence for many years and has been attempted with other types of infection. But as we know from the data that has been released, this is a highly effective vaccine. I'd like to take the opportunity to show you why I think this is very safe. So first, understand that what has been done is the coding for the particular mRNA, messenger RNA, for the proteins that are located in the spike. When you look at the shape of the coronavirus, it's a sphere, and there's little projections that come off it, and these are where the virus attaches to the target cell. And in this particular case, the delivery system is a lipid nanoparticle, which encapsulates the mRNA. And the reason for that is that it'll facilitate the mRNA remaining in the cytoplasm just long enough to code for the target protein. And all this is done in the cytoplasm. The material does not enter the nucleus. It's not incorporated as part of the genetic makeup of the cell. And when this is injected into the deltoid muscle, it's taken up by the myocyte. And the production of these spike proteins are then created. It's much like if you think of, if we gave naked mRNA, the RNase would degrade it so rapidly that you'd have to use enormous amounts of it, making it a unwieldy and impractical type of vaccine. And once the spike proteins are synthesized, in the cytoplasm through the normal protein synthesis pathway, it then becomes available to the immune system, and hence antibodies would be made. My colleague, Dr. Dan Johnson, who couldn't be with us tonight, makes an analogy. May I have the next slide? with M&Ms. Can you go back? One more slide. Can you go back one? With M&Ms, where the idea is that the candy will melt in your mouth and not in your hand. So think of the candy coating as being like this lipid membrane. May I have the next slide? So in addition to generating different globulins, the antibodies to these proteins, we also have a cell-mediated process that's going on. May I have the next slide? And you can see that these same proteins will be recognized by the cell-mediated immune system in addition to the humoral immune system. And this slide demonstrates whether it's the CD8 T cells, B cells, and eventually the CD4 cells will be also involved in an immunologic response, thereby building what we know as longer lasting immunity against these proteins, which would in fact inactivate the ability of the virus to attack the cell. And that's really the strategy behind it. And as you're going to see from the data that Dr. Chundy is going to share with you, it's highly effective. So we can go on to the next slide.
Thank you, Natalie. Can you get to the first slide? So the Chicago Medical Society recognized early on when we did a survey that there was a significant amount of hesitancy, especially amongst our own membership, physicians, for taking the vaccine. They had a lot of questions, whether it's safe, whether with the correct, how the vaccine worked, and correctly so, because there was a lot of misinformation and lack of information. Now, with over 16 million doses, we do have a significant amount of information. And so our goal was to make not just physicians, but also everybody in the community that we think would help take the vaccine hesitant people and convert them into the vaccine acceptance people. This includes celebrities, social influencers. It includes baseball players, basketball players, sports folks, as well as the clergy, the religious leadership, to become trusted messengers. So they become effective communicators about the vaccine. So we wanted to understand about how mRNA vaccines work and be comfortable communicating with your patients as well as constituents. So they are very comfortable saying that the vaccine works and it's safe. I think we're underselling this vaccine. This vaccine is 95% effective. If you ask the infectious disease community out there, before all of these vaccines came out, what percentage would you take as far as vaccine goes? I think people would have been happy with 70, 75%. If you take the flu vaccine, most of the time it's 50, 60% effective at best. This vaccine is 95% effective. In fact, a couple of my close colleagues who have significant immunocompromising states and took the vaccine despite being on steroids, despite getting biologics, I asked them to test their antibodies because I wanted to see in this significantly immunocompromised folks, would they get antibodies? 100% of the people I've had tested have gotten antibodies. So the vaccine really works. And they've been about 6. It's not like I have hundreds and hundreds of patients, but 6 people. So it does work. I want everyone to become like an expert and be armed with knowledge to explain the myths and the fallacies to convert the vaccine hesitants to the vaccine acceptance. I think from a Chicago Medical Society, if we can accomplish this, it would be a nice way of getting 100 million people and eventually 340 or 350 million Americans vaccinated. So we can all go watch the White Sox win the World Series and eat a hot dog in the ballpark. Next slide. So one of the things that people say is, well, we had a lot of conversation about getting natural infections. You know, people had COVID parties to get infected. There've been fatal consequences of some of these COVID parties. And I wanted to bring up 2 cases. One case I just saw a week before, and this other 42-year-old gentleman who I took care of several months earlier. And this gentleman had 8 days of fever, cough. He was a well-controlled diabetic. He quickly progressed, had required a face mask and a BiPAP. eventually was placed on a ventilator and received extracorporeal membrane oxygenation. And this was complicated by chest tubes, and eventually got a lung transplant, which is a tremendously difficult transplant and alters his life forever. And that's from natural infection. This other young lady came in, was admitted to the MICU with low blood pressure, headache, and shock, and for all practical purposes, looked like she had toxic shock syndrome. She had abnormal liver enzymes, as you can see, with a bilirubin of 5.1, and she never knew she had COVID. We tested her repeatedly with PCRs; they were negative, so she didn't have active COVID. But one of my partners tested her with an antibody. The antibody came back positive, and we thought she had a multisystemic the inflammatory syndrome with COVID, which is like Kawasaki's. She'd received steroids and she got much better and was sent home. So this is not a benign illness, and even young people get very sick, not just the old people. Next slide. So natural infection in any infectious diseases is more deleterious to your health than getting a attenuated infection from immunizations. When Jenner started with the cowpox, there was actually at that time an anti-vaxxer campaign. There was a very famous poster I didn't bring because for lack of time, but it has basically people growing cows out of their arms and everywhere else where he scarred people down with cowpox to prevent them from getting smallpox. So there were anti-vaxxers back then. The current anti-vaccine campaign is from a gastroenterologist who incorrectly and since he's lost his license in England, tried to put measles as the cause of getting autism, and he's been disgraced. So vaccines are always safer than herd immunity by natural infection because the side effects of natural infections are significant, and that's why vaccines have to be very, very safe. Whereas if you're treating an illness, like if you're treating an endocarditis like we treat, or a brain abscess, we can give antibiotics, have significant side effects, and kind of push through it because the medications are treating an actual infection. Here you're giving a vaccine, an intervention to a healthy group, and so you must have very vigorous trials. And so next slide. So the mRNA vaccines absolutely underwent the same rigorous trials as every other vaccine that's been approved. The vaccine with the shortest trial period from the time they conceived it to coming up to the vaccination is mumps, which was about 4.5 years. So this vaccine was done in rapid time, and not because they changed the safety, but because the government guaranteed essentially that the drug companies would not be on the hook for losses if the vaccines didn't work. In fact, we had several vaccine companies where the vaccines did not work. So what does the mRNA vaccine do? It basically takes messenger RNA, which is the RNA that gives your ribosomes the instructions to make protein, in which case the protein that they're making is for the spike protein, parts of the spike protein, and each of the vaccines are different for which parts they can make, so you can't interchange them. And it starts cranking out the spike protein in large amounts, in which case your body then says, hey, I've got an invader, and then starts making antibodies, and these antibodies then get presented to antigen presenting cells, and then eventually you end up getting T cell immunity and B cell immunity, and it's a long-lived immunity. So that's how the messenger RNA works. The problem is when we tried to use messenger RNA before, and the development has been going on for about 25 years, they gave naked messenger RNA into muscles, so where you needed large quantities and caused severe reactions. So the Moderna and these BioNTech vaccines were actually made to fight Zika, and the secret sauce that they have is that lipid, this what's called polyethylene glycol, the pegylated stuff that you take when you take Miralax or something like that, to transport it into the antigen presenting area. So this is a tremendously important breakthrough of how to get this RNA into areas it's supposed to go. Next slide. So how were these vaccines developed? They didn't happen overnight. They were actually developed for Zika. Remember the virus that caused all of the birth defects in Brazil and all over the world. And so two companies thought this was a great thing that they needed to do, Moderna and BioNTech, and develop these RNA vaccines against Zika. And as they were ready to inoculate the world against Zika, Zika just disappeared. And it disappeared because the vector, which is the Aedes aegypti mosquito, was carries the same vector for dengue. And dengue had actually been around for a long time, which is another viral illness. And the mosquito vector, the dengue overtook the mosquito vector and displaced Zika in the hindgut. And so Zika had no place to reproduce and kind of died out. And so these people were left with a lot of vaccine and no people to take it. And then lo and behold, you get SARS-CoV-2 causing COVID-19, and they had everything developed except the antigenic portion. The Chinese came around and gave to the world at that time, when COVID-19 came out, the genetic sequencing of the whole virus. And the NIH within a week knew which part of the of the spike protein was the most antigenic portion, and gave it off to these companies. And these companies came up with a vaccine within 6 weeks, literally 6 weeks, to get it out to trial. And then the trials were done at the same time that production started, because they bet on this. The government bet that this would work, and the NIH had been using Moderna. BioNTech was bought by Pfizer, and Pfizer decided not to take any federal funding. And so Pfizer put their money where their mouth is, and fundamentally took care of the vaccine. So the federal FDA approval process for safety was unchanged. Next slide. So what is the safety? You can see for the Pfizer vaccine, 43,000 participants were followed up at 8 weeks. Moderna, 30,000 participants for 8 weeks. There was no major side effects, no anaphylaxis, no no paralysis, no Guillain-Barré. They did have 2 anaphylactoid reactions in Britain after the vaccine came out. Now we know a lot more about this. 16 million doses have been given. We know we have a good idea what the side effects are going to be. For the Pfizer vaccine and the Moderna vaccine, there's about 95% efficacy. Next slide. So in the last week, we've had 16 million doses given. There are a fair amount of anaphylactoid reactions. Nobody has died of anaphylaxis. And this happens to be from the polyethylene glycol chain. And the people who get this are people who have mostly prior history of reactions to severe reactions to vaccines. There have been a few who have not had reactions. And so it's recommended that you keep the patient for 15 minutes if they have no reactions. And if they have prior reactions, keep them for 30 minutes. There has been no Guillain-Barré yet reported, and no transverse myelitis. There are reports now of small fiber neuropathy, which goes away. And then there's numerous what they call minor side effects. I had both doses of the vaccine. It does, and I already had COVID, so I had antibodies. And it does knock you on your butt when you take the vaccine. You get chills, you get headaches, you get body aches, and this occurs more often in the younger, healthier immune systems. The people over 65 and 70 who get it as their immunity wanes don't have as much reaction. And this is borne out within our own group, where the people younger than me had more reactions. But it goes away. It's self-limited within a day, and it's tolerable. You take some Tylenol and you're okay. The other strange thing we've noticed, and Dr. Paul Offit, who is like the guru of vaccines, he's in Philadelphia. You can watch his podcast; they're excellent. We're seeing these delayed reactions over the arm, and it's basically a redness and tenderness over the arm. It's not like an arthros reaction or an immediate type of reaction where it happens within 12, 24 hours. This is happening 7, 8 days later. There's redness, there's tenderness over the site. He feels this is CD4 mediated along with the CD8 cytotoxic T cell response. And because you have so much of the mRNA being produced in that area, you're developing a local response, and it's self-limited. It goes away. There's no major side effect from that. Next slide. So the 2 vaccines are 2 different vaccines. The Pfizer vaccine requires ultra cold storage, so minus 70 degrees. It's stable for 6 months. It's good for 5 days at 20 degrees. So once you take it out of the ultra cold storage and put it into your regular freezer, it's good for 5 days. And after you reconstitute it, it's good for about 6 hours. So you have to use all those doses within 6 hours. It's about 30 micrograms on day 1 and day 2. Going in, you really don't feel it at all. It's such a tiny dose. Moderna is minus 20. which is most doctors' offices have that and pharmacies have that. It's better for rural areas and private doctors' offices, and both are fairly well tolerated. Next slide. So they're very similar in comparison. So the local reactions that I talked about when you get it, if you've taken the vaccine, are they hurt. Your arm hurts where you get the shot. Okay, and the first shot, and then there's regional lymphadenitis, especially in the axillary area, and when I did not have adenitis, but several of my partners had significant adenitis where they received the vaccine. Bell's palsy was reported in four of the patients and 0 in the placebo. But interesting, the Bell's palsy is no higher than what you'd expect in a background of Bell's palsy. One of my partners previously had Bell's palsy. She took the vaccine. She didn't have Bell's palsy, so it doesn't seem to be associated with Bell's palsy. The Moderna vaccine recently in California, one of the batches was halted because there was a significant amount of reactions with one of the batches. And then they're resuming the use of Moderna. So these vaccines do have local side effects like this that you should be aware of, but the systemic side effects of Guillain-Barré, of transverse myelitis have not occurred. Next slide. So what are the indications? You cannot mix these two vaccines together. They target different parts of the spike glycoprotein. The Pfizer is indicated for ages 16 and older. They tried it on the younger people. Right now, there are trials going on in pediatrics. The Moderna vaccine is indicated for 18 and older. The dosage we talked about is 30 micrograms, and it's given on day 1 and day 22. The Moderna is 100 micrograms given on day 1 and day 29. The efficacy is 95%, as I've said over and over again. The difference is the Moderna in ages of 65 and over, the trials there showed the efficacy is a little less, 86%. And there's no difference in efficacy based on race. These trials had excellent representation of race. They had over 10% of African Americans. They had appropriate amount of Latinx participation, a good amount of Asian participation, and so a very nice mix of what America is. So this is unlike prior trials, which were predominantly Caucasian participation. So the protection starts for the Pfizer vaccine about day 10 after the first dose. The efficacy before the second dose gets up to as high as 70%, and it's less likely to cause headaches compared to Moderna. The Moderna vaccine gives you protection about 70 to 80% within 21 days, and it protects against severe disease and death. And this is really, really important because the type of immunity you get is neutralizing antibody immunity. So you get good neutralizing antibodies for both of these vaccines, and it prevents against death, respiratory failure, and all the horrible things that you're seeing with this virus. It does not give you, as of yet, sterilizing immunity. And the difference is in sterilizing immunity, if you have sterilizing immunity, you won't transmit the virus. You won't be able to carry the virus in your nasal cavity. So you still have to wear a mask. You still have to do the appropriate social distancing until we get lower levels of community onset of COVID-19. So it doesn't prevent nasal carriage of virus. Next slide. So these are from the trials you can that Pfizer had, and you can see on this side here with the Pfizer vaccine on about day 8 you start seeing the separation from placebo, and the efficacy by day 28 gets up to about before you get your second dose about 80%, and after the second dose within a week you're up to about 95%. Within 2 weeks you have optimal immunity. The Moderna vaccine takes a little longer to start separating itself as far as the efficacy goes, but by day 28 and you get your second dose, you really start getting good immunity for the virus. So they're really nice vaccines. Next slide. So what are trusted messengers? We need to use the correct messengers. So people act when they trust the messenger. And this is not just for vaccines. It's also important for everything else you do in your life. Well, as a physician, if you're a pastor, as a pastor, you must have the trust of the people that you're talking to. The most effective messengers are experts and trusted leaders in our own communities, both geographic ones and digital ones. People are on on the internet and communicating as we are. Examples of trusted messengers are physicians, school nurses, pastors, community leaders, and in African-American community, barbershops become a local area of conversation. So you really want to convert these local area of conversations to the right message and get the correct facts out there. And context is really, really important. Political context, social context, and experiential context. Today, when I'm talking about my vaccine that I took, I'm trying to give people the experiential context of, yeah, it's not a walk in the park where you don't feel it, but it's not the end of the world. It's much better than getting COVID. And peer pressure really does work. Abhijit Banerjee won a Nobel Prize for this on how people shop and how people do things. And peer effect is real and it works. Getting the word out, being the agent of change, really makes a difference. So everyone should be talking about taking the vaccine rather than finding a reason not to take the vaccine. I think we've been underselling this vaccine terribly. This vaccine is a great vaccine, 95% efficacious vaccine. It has some minor side effects. You have to take 2 doses rather than 1, but compared to getting COVID and the fear of transmitting it to everyone that's around us, this is a tremendous achievement. I mean, this is a scientific achievement of like going to the moon, essentially, so quickly, with so many people putting efforts into this. And this is a long-term effort. This is not something that was just immediately thought up of, as we talked about. So next slide. So Dr. Lynn is going to talk about pregnancy, lactating, and there were questions about sterility and stuff like that, which he's much better at talking about than I am. So Dr. Lynn.
Thank you very much, Dr. Chandi. May I have the first slide, please? I want to, again, emphasize the importance of vaccination as a public health achievement. And we're talking about multiple diseases. And for those of you who are dealing with reproductive age women, this is a discussion that we have all had with our patients on the importance of being up to date on their vaccinations. Because regardless of the vaccine, having the target infection tends to have much different consequences. I'll start off by making a parallel analogy to influenza. We actively and aggressively try to counsel our patients to accept the influenza vaccine while they're pregnant, because influenza in pregnancy can be a very severe disease. For those of us who had experience with the H1N1 pandemic that occurred a number of years ago, this was when we found pregnant women in the intensive care units. In some centers, they even had to resort to ECMO, and there were deaths. Now, another thing that I want to emphasize, and just as a point of review from what we just covered, the mRNA vaccine is not a viral genome. It is not an attenuated virus. It's not like rubella or zoster, where we would give a vaccine that would have a different reaction. And please, when you think about a vaccine, in the case of these COVID-19 vaccines that we're speaking about, it is not inducing a systemic illness. It's inducing an immunologic reaction to a very specific set of proteins. We realize that there's a lot of misinformation, a lot of anxiety out there amongst our patients. We do not have level 1 evidence because pregnant women were not enrolled in the studies. Lactating women were not purposely enrolled, but we do know that women became pregnant, and we do know that women do breastfeed, and I'm going to be covering this in a moment. But again, I want to emphasize that these vaccines do not cause the infection. So when we're comparing the risks and benefits, you have to compare the risks, which we know are very, very minimal, compared to the risk of an infection, which has reproductive consequences. May I have the next slide, please? So these are the recommendations that the American College of OBGYN and the Society for Maternal Fetal Medicine and the CDC have put forward, and that it's recommended that COVID vaccine should not be withheld from pregnant individuals who meet the standard criteria based on the ACIP committee's recommendation for priority groups. So if you're a healthcare provider, you'll be group 1A, and you should be getting the vaccine, even if you're not, even if you are pregnant. And subsequently, as we go down to 1B and then eventually 1C. As I stated before, we can't say that we've actually have a group of women who are actively vaccinated while they were pregnant to be included in the study. But we do know that these cases are accumulating because many women may not even know they were pregnant at the time that they were vaccinated. But there is no data to indicate that the vaccines are contraindicated. And there are no safety signals generated from the DART studies for the Pfizer-BioNTech vaccine that we should withhold it. Now, pregnant women are individuals that should be free to make their own decisions in conjunction with their clinical care team. And it is very important, and this kind of goes to the idea of the trusted messenger, that patients should be counseled and have a discussion with their providers. May I have the next slide, please? So one of the things that we have to look at in helping the patient make this decision is the level of activity of the virus in the community. So if your patient has to be living on the island of Tarawa in the South Pacific, where that particular government closed down travel, and right now there's no reported infection there, and they should be maybe a little more comfortable that there's not a lot of urgency for them to become vaccinated. But when we're talking to people in Chicago, or many or most of the metropolitan areas in the United States, this infection is occurring, and it's occurring with a significant degree of frequency. And our recommendation would be that they should consider being protected. And we already talked about the potential efficacy of the vaccine. It is really outstanding. Now, will it be as effective, let's say 95% in a pregnant woman? Because we do know that there is a subtle blunting of the immune response in pregnancy. We can't say that, but we do know that it should be highly effective. Most importantly, we have to look at the risk and potential severity of maternal disease, including the effects of the disease on fetus and newborn. We do know now from many case reports that people who get severe systemic disease may have preterm labor, and this can be, though, a significant illness in these people. And as we said before, we don't anticipate any safety issues from the vaccine because of the nature of what it's doing physiologically. Again, we don't have a virus. It's not like we give a attenuated virus, which induces a viremia that may not be clinically severe, but that potentially could affect the mother or the fetus. My next slide, please. Now, similarly, in lactating individuals, the American College and the other groups have said pretty emphatically that these women should be offered, who are lactating should be offered vaccination. Again, using the same criteria. Same thing, they were not included in most of the clinical trials as a target population to be studied. But these vaccines should not be withheld because we see no physiologic reason that this should be a threat to the baby. Now, there are theoretical concerns that we always can raise. We're very good at raising theoretical concerns as physicians. We have very, I'm going to say, rich background to be able to think these things through, but they do not outweigh the potential benefits. And so we don't recommend that if a woman becomes vaccinated, that she discontinue breastfeeding either. Next slide, please. Another important group are women who are actively either attempting to become pregnant or contemplating pregnancy in the future. So unlike rubella, where if we discover that we have a patient who's not pregnant, who's anticipating pregnancy, and we test for antibody and we determine that it appears that the patient is not immune, we recommend vaccination, but we recommend deferring or delaying pregnancy or attempting pregnancy for at least 3 months after the vaccination. This is not the case with the COVID-19 vaccine. It's strongly encouraged that people who are not pregnant become vaccinated because getting that immunity will then protect them once they do become pregnant from a much more significant disease. And we also recommend completing the vaccine course and not truncating it because you're trying to get pregnant or you become pregnant at that point. There have been some isolated reports in males that the COVID infection, this is males who have recovered, among many systems that could be affected because we think is the inflammatory response. It appears that there may be a reduction in sperm count. But this has not been reported to my knowledge from the vaccine. Again, you're not inducing a systemic illness. You're inducing a localized immunologic reaction. May I have the next slide, please? Dr. Chandy, back to you.
Oh, back to me. Thank you. Next slide. So there were some frequently asked questions that we get on a regular basis. So the first question is, if I had COVID, do I need the vaccine? And I had COVID and I took the vaccine. And the reasoning is COVID natural immunity wanes. This mRNA immunity seems to last a lot longer. Not everybody who gets infected with COVID-19 develops antibodies. People who are severely ill, 20 to 30%, 25% may not actually develop antibodies. So taking the vaccine ensures that 95% of the time you're going to get immunity, which is very, very useful. The second question, which is asked, actually the most frequently asked question is, will the vaccine alter my DNA? And as Dr. Lin elegantly pointed out, the RNA does not enter the nucleus and cannot alter your DNA. This is one of the big internet things going on right now, is that your DNA is going to get altered with the RNA vaccine. But as you all know, RNA just makes protein in the ribosomes and the cytosol. It does not enter the nucleus and does not alter your DNA. The other thing that's in the internet mimes is that the vaccine actually has some sort of chip that the QAnon people, like folks, are saying the chip is being inserted, and the Gates Foundation, Bill Gates, and all these folks are tracking you, and that's absolutely a false myth. So that's just crazy stuff on the internet. So will the vaccine make me sick with COVID? Will the vaccine? No, the feverish feeling and the way you feel. There were several questions about feverish chills, body aches after vaccine. It's basically your immune system is primed. It's running like a Porsche, and it's waking up and it's making a cytotoxic T cell response. You're releasing cytokines, and you're not feeling well. So take some Tylenol. Usually self-limited, usually about a day. The second dose, you've been primed, and the second dose may be a little bit worse. So one of the problems is, a significant number of people are having to take off work after the second dose. So be ready for that. Don't sit here, and if you're running a company or you're managing a unit where you need all your workers, don't vaccinate them all on one day. Stagger it. Vaccinate people on Thursday or Friday, so they have the weekend off. So that's important. If you're an airline and you're running it, you want to vaccinate all your pilots at the same time. You may not have a whole bunch of people flying the next day. So it's important that you kind of stagger the vaccination, because it takes about a day after the second dose to feel better. And then will the COVID test will the vaccine turn my COVID test positive? It does not turn your PCR positive. The polymerase chain reaction that you do with the nasal swabs are do not turn positive, because all you're testing for, all you're giving in the vaccine is a portion of the spike protein, not the portion that the PCR is testing for. It may turn your antibody positive. It should turn your antibody positive, but not all the test for these antibodies. You have these little lateral flow assays that people use that look like pregnancy kits. Those are very insensitive and a lot of cross reactivity. So it will not, that they may not work. But the blood tests that people do with the antibodies, they do turn positive. Next slide. So why do I need to wear a mask if I had vaccination? This is a common question that today actually got a long meeting about this. What do we do with healthcare workers who've been vaccinated? Do they need to wear? You absolutely need to wear a mask. And I touched on this earlier. The vaccination does not give you sterilizing immunity. Sterilizing immunity means you get vaccinated and you cannot get the virus into your nose or anywhere else. It gives you neutralizing immunity. So the vaccine basically prevents you from getting infection into your lungs and other organs, so you don't die from it and get really sick from it. So you can still carry it in your nose. So as long as you have a significant amount of prevalence of infection in the community, you must wear a mask until the rates are really low, and then we can be mask free for a period of time. But what is the one thing that you've all noticed if you've worn masks? I have not had a cold since COVID started. I've been scared out of my mind of getting really bad COVID, but I have not had a cold. And that's been the observation throughout both hemispheres, the southern and the northern hemisphere. The lack of influenza because of COVID, mostly by wearing masks, and the lack of having all these upper respiratory infections that we suffer from in the wintertime. So masks do work, which is a change in paradigm from the infection control world, because we didn't believe you needed to wear masks all the time in the wintertime. In fact, we would fine people and tell them that you're violating our CDC guidelines by wearing masks in the hallway in the hospitals. But now we see that the masks really do work. So next slide. I just want to touch on an important development because people are going to ask you. So it was all over the press about these variants, and these variants are significant. There's two variants, the UK B117 variant. And this variant started in the UK probably September, October. And it basically came from one lineage. And it fundamentally has taken over the United Kingdom and caused significant amounts of COVID-19 for an outbreak, because it's much more transmissible than the virus that was out there. It's about, they think, 40, 50% more transmissible. I think the data is not clear on that, maybe as low as 20 or 30%, but that's a significant number of people getting infected. And it's fundamentally no more, it's no more virulent, but because more people are getting infected, it's overrunning the hospitals. Both these variants share the 501Y mutation, and it's in the S gene, which lies in the receptor binding domain. And why is that so important? This is where the spike protein attaches to the ACE receptor. And that's where the antibodies are bound to the virus. And that's where also the vaccines work. The South African variant has two more mutations, more than the 501Y. They have E484K mutation, and that's a pretty important mutation, and the K4-117 mutation. And the E484 mutation has three little amino acid changes. One of them turns the receptor binding protein about 20 degrees and actually gives a tighter attachment to the ACE receptor. So the virus attaches tighter to the ACE receptor and makes it hard, and it actually makes it harder for the virus. It actually makes it easier to transmit that way. The second change is in the charge. Before, at the receptor binding domain, there were two negatively charged amino acids. So they kind of repelled each other, kind of flapped in the wind. Now you have a positively charged amino acid and they bind tightly. So both of these, this combination of these two changes allows the South African variation to be more effectively transmitted. And much more importantly, there was a study published this Saturday in preprint version that looked at this virus and how it works against in people with convalescent serum. And they found that it does not bind as well to the convalescent serum and monoclonal antibodies. So this actually may be an escape variant that eventually, if it continues to evolve like this, will become a vaccine escape variant. Uh, it has to vary much more. Right now, the vaccine works against both mutations, and it's really important. Time is of the essence to get as many people vaccinated as possible, because then you'll have much lesser amount of virus being circulating, and then the amount of mutations decrease, and hopefully the disease will die off. I don't believe that's going to be the case. I think we're going to have this being a long-lived infection amongst humans, and it's going to become like the common cold eventually. But right now, the hope is we can get enough people vaccinated so the mortality rates fall down, and you can decrease transmission enough where the best case scenario is what happened to SARS-1, where it kind of died off. This is going to be difficult to do simply because there's so many people infected. Next slide. So what do we know? So we know the vaccine is safe. And I think this is key for everyone to know. This is a safe vaccine. There are side effects. There are local side effects. There's all this anaphylactoid reactions that people get, but these are all manageable things that we should be able to handle. It has a complex cold chain, which is hard. The Pfizer vaccine requires really cold -70. The Moderna vaccine requires -20. So it's very difficult to use. It's very difficult to use in developing world. 2 doses, which makes everything more complex. You got to bring people back 21 days for the Pfizer, 28 days for Moderna. And they can't be mixed. You can't take Pfizer one time and Moderna the next time. And the total number of doses is about 150 million people can be given. So you had to buy 300 million doses of vaccine, which was done by the prior administration. The Johnson & Johnson vaccine, which will likely be approved next month because it looks like it's a very good vaccine, it's a single dose vaccine. It is not a true mRNA. It is not an mRNA vaccine, and there's about 150 million doses of that vaccine also bought. So we should be able to vaccinate all of the United States by the end of the year if we can get our act together. And it looks like there's a federal plan out there to do this. Next slide. So some of the things we don't know. How long will the vaccine protect you? We know it's going to protect you for at least 5 months now, because it's how long the testing's been out. We think it's going to protect you for at least a year or longer. Will the vaccine prevent transmission of infections? The belief is it does prevent. So there's studies out there that look at viral loads in the nose after vaccination. And so the viral loads are less in people who are vaccinated than unvaccinated people. So the lower the viral load, the less the risk of infection. So it should decrease the efficiency of transmission, and that all important R naught, which is a reproductive number, if it drops below 1, the epidemic and the pandemic will die out. And that's the hope. What do we don't know that we're going to find out soon is use in children. So the vaccine trials in children are ongoing. And so that's something that's going to come out soon. Use in pregnancy and lactation. And Dr. Lin talked quite nicely about how we shouldn't withhold the vaccine in pregnant and lactating women simply because the natural infection is much worse. The long-term side effects are yet to be worked out. I was just looking at the VAERS database on the Pfizer. There's like 5,000 reports of adverse reaction to the Pfizer vaccine thus far. There's been about 10 million plus doses given out. So that's not too horrible. And basically, it's a subunit vaccine. So you're not going to get most of the reactions which will occur within 6 weeks. You're not going to get long-term, years out sort of reactions. Next slide. So what don't we know? All the long-term side effects. One thing people are worried about is Guillain-Barré because of swine flu in the past. So natural influenza, in natural influenza, you get about 1 in a million people will get Guillain-Barré. with the I'm sorry, with the vaccination, you get 1 in a million. With the natural influenza, you get about 17 cases in a million. So still, influenza vaccine is safer than getting natural infection. That's what we've been talking about throughout. I think that's it. Next slide. And we're good. So I'd like everyone, we'd hope that everyone would join our trusted messenger, and there's the website. We will have a slide deck, which I promised last week, but we should have it done within the next few days for everyone who's forward facing so they can go talk to their communities about this. Be more than happy to answer questions. And we do have many more people who are experts on vaccinations that can answer questions if you email or send it to us. This is something that the Chicago Medical Society would like to do for the Chicagoland area as well as all of Illinois, because we'd like to get through this as quickly as possible. And as a medical society, I think this is one of the important things that we do. So I'll stop there. And there are several questions on the chat, which were started. And one of the questions was, should a patient who gets a side effect from the first dose of Pfizer vaccine with muscle aches, headaches, get a second dose? Could the side effects of the second dose be worse than the first? And the answer is yes for both. You should get the second dose. Yes, you may have worse reaction, but that could be easily managed with Tylenol, little warm compresses over the arms. This is a question for Dr. Lin. I've heard of reports of decreased sperm quality after natural infection. Any indication on fertility after the vaccination? Dr. Lin.
Yeah, I'm not aware of any. Realizing that this can take a very long time in order to determine that. I think this is something that will be more forthcoming. Biologically, I don't see any reason why, unless this immune response causes a direct attack on the spermatocyte, and I can't think of a plausible reason why it should, but more will come as we have much more experience with it. But I don't think it's a reason to avoid vaccination.
And then there's a question about any comments about non-mRNA vaccines. Yes. So there's several vaccines out there all over the world. The Chinese have made a whole spike glycoprotein vaccine. The AstraZeneca vaccine, which is also on the docket to get approved, is a chimpanzee vector virus vaccine with spike glycoprotein. These are both to be the AstraZeneca vaccine is not as efficacious as the mRNA vaccine, but much easier to give. It doesn't require complex cold storage. It also requires 2 doses. A smaller priming dose, the lower dose does a priming dose, and a regular dose is a second dose. It has not been approved in the US because the way the studies were completed were not consistent. They changed the protocol in between. India is planning on doing 3 million doses a day of this vaccination, and it has been found to be safe over there. The Johnson & Johnson vaccine is another a viral vector vaccine with a whole spike glycoprotein. It's found the efficacy in preliminary reports. There was just a New England Journal article published a couple of weeks ago shows it's very safe vaccine, and the efficacy seems to be excellent. So you'll now have a single dose vaccine coming out to complete the circuit for how many, and all of these vaccines are going to be needed if we're going to vaccinate everybody in our society. And we need everybody to think about vaccines as being safe. I mean, we're as physicians think we're saving all these lives all the time, and we help people individually. But the greatest things that have helped mankind have been improved economic growth, okay, which helps people get better health-wise. Toilets and indoor plumbing, which prevents mass outbreaks of diarrheal illness, because in the developing world, the child having 3 episodes of diarrhea within the first year of his life, their mortality is very high by the time they're 5. And vaccinations. Vaccinations are the vanguards of a organized society with large cities, because you prevent all of these massive outbreaks of cholera, of typhoid, not cholera so much, because cholera vaccine is not horribly effective. Of measles. Measles wiped out the Native Americans in the North America. So vaccinations are absolutely the cornerstones of society, and we should not be taking these the postmodern society that we live in which is a distrust of government, distrust of experts, is what we're running up against. And so, how do we fight that? We fight that with appropriate knowledge, talking about what we know, and bursting the myths out there that are out there. And there's a very organized group of anti-vaxxers for reasons that are unbeknownst to me why there would be anti-vaxxers. And then let's see, does the vaccine cause a positive antigen test? No, it should not cause a positive antigen test. It doesn't induce antigens. It can give you an antibody test that are positive. Can the vaccine result in a positive COVID-19 test? We've answered that. It does not give you a positive PCR. And then should I do an antibody test after the second dose of the vaccine? And the answer is no, that's not recommended. And the only reason why I ordered that was one of my nurses was on a massive dose of solumedrol, like a gram of solumedrol. And I was wondering if that ablated her T cells. And it did not, because she actually developed an antibody. So that was a nice thing. It gave me reassurance that this vaccine really does work really well. And so that's a very good thing. If I get vaccinated, can I be free from quarantine? That's exactly the question we answered during one of our meetings today. As an infectious disease doctor and a person who sets policy in a lot of the hospitals that I go to, we've decided not to quarantine anybody if they've been vaccinated with 2 full doses and have completed a week after the vaccination, because by definition, you are immune. It doesn't say you should not, you have to wear a mask, you still have to follow the normal protocols, but you don't need to be quarantined anymore, which is a big deal. You're eventually, what's gonna happen, you can see it playing out already, in like Qantas Airlines is requiring a COVID test or vaccination before you get on the airplane. And this is gonna happen more and more in private businesses. And so it's really important to get vaccinated. Any concerns for patients with pre-existing autoimmune, autoinflammatory diseases with any of the vaccines, mRNA or adenoviral vector vaccines? Actually, the answer for the mRNA vaccines is absolutely not. These are particles of protein, and pre-existing autoimmune and autoinflammatory diseases should not cause any issues. If you have previous history of anaphylaxis, then you worry and you should be closely monitored and get the vaccine in an office that has the access to resuscitative services. As far as the adenoviral vector vaccines, the original one was a chimpanzee vaccine for HIV. It was a human adenoviral vaccine, and it was found that most of us have adenoviral antibodies, and so the uptake wasn't good, but they started using a chimpanzee adenoviral vaccine, which is what the AstraZeneca vaccine is. There are more questions. And do we have adequate personnel available to give a vaccine? And the answer is no. If we're going to vaccinate Chicago, the Chicagoland area, which is about 6.5, 7 million people, you're going to need thousands of volunteers out there, which means doctors, nurses, EMTs, dentists should be able to give vaccines, veterinarians should be able to give vaccines. And so we're looking for volunteers so we can have a group of people out there. We're actively we're actively trying to get help out the city in their vaccination efforts, so we can have a pool of people to go help them with vaccination. So all of this is needed. Everybody has to pull together on the same way to get this done.
And let me just interject, as many of you are aware, one of the plans, it will be to actually use the National Guard to assist in this activity too. And we can't expect hospitals to bear the burden of this because they're in a different business. They're not in the vaccination, large population business. So none of these things are being worked out, but certainly the people like yourselves who are on this, participating in this, we intend to reach out to you to find out if there are people willing to volunteer, whenever the need pops up. And we are looking at and trying to assist in this effort, and are in negotiations about creating I'm talking about the medical society in terms of being able to assist in the creation of some mass vaccination center.
Thank you, Dr. Lynn. There's a question about if you have a monoclonal antibody, what's the earliest, when can you receive the vaccine? 90 days, because you now have a passive antibody which may block it. So after 90 days, whether you've had plasma, whether you've received monoclonal antibodies, then you can get your vaccination. If you've received a dose of vaccine and then you turn positive in between, then the you complete your dose of the second vaccine. There's a question here about receiving a Moderna vaccine on January 6th, had some pain in the arm for 2 days. And then what I talked about on January 20th, developed a patch and a rash on the same arm. This is something we've actually observed quite a bit. And Paul Offit talked about this being a CD4, CD8 interaction, a cytotoxic interaction. And so you should get a second dose because people do convert to IgG. The duration is not known if you don't get the second booster dose. So you should get your second booster dose on February 3rd. And I think we're running out of time, so we'll answer these questions after this is all over. There's one question by Dr. Levy. I have a right-wing patient who told me he would not get a vaccine because of some component that is derived from human abortus. Please refute. It is not derived from any human abortus. The original mRNA stuff was from, I believe, some fetal material, originally the fetal cell lines.
Some stem cell
fetal line.
And fetal stem cell, but that's not what is currently being done. This is not how the vaccine is being produced. It's not grown in tissue culture. It's not like other vaccine are grown in eggs. It's because it's not a virus. It's actually, think of it as the code is being built in the laboratory, and it's being coated with this lipid polymer, and that's what it is. So it's a different animal than what people have been attributing to it.
There's a question about, I attended Kane County meeting yesterday, and I believe they said if you were vaccinated and are exposed to COVID-19, you should still quarantine. From an infection control perspective, that doesn't make sense, because if you took up the vaccine and you're asymptomatic, I'm talking about asymptomatic people, not people who are symptomatic. If you're asymptomatic and you wear a mask and you carry on, you should be fine. So I don't understand the reasoning of quarantining people who are vaccinated.
And again, we're emphasizing that you complete the full vaccination course and give adequate time, post the second inoculation. If you're getting one vaccine and now you acquire the one dose and you acquire the infection, yes, you're not immunized yet. So I think it's a different thing, and I'm not so sure what was discussed in Kane County.
Yeah, and then how long the vaccine is going to be effective for. The infectious disease world believes it to be effective for a year, but if the virus mutates rapidly, and this is not a rapidly mutating virus, this is a beta coronavirus, you usually get 1 or 2 mutations a month. But then suddenly they had a blip where there was 23 mutations from one single person in South Africa, 22 mutations from a single person in Britain, which changed the paradigm. So that's the issue. In our institution, we're being told not to get vaccinated within 90 days post-vaccine. If you look at the CDC website, that was based on the concern about how much vaccine that's available. The CDC website says that if you're asymptomatic after 2 weeks, you can go ahead and get vaccinated after infection. The issue is you may have an enhanced reaction based on antibodies. So you get ready for that. It does get you a little bit sicker if you already have pre-existing antibodies. So I think we'll stop there, and I'd like to thank everyone for participating. And one last question: I was told that because you were vaccinated does not mean you cannot catch the virus and spread it. That's absolutely true. It's because of neutralizing antibodies versus sterilizing antibodies. This vaccine does not give you sterilizing antibodies. So you still need to wear a mask until the incidence of disease is very low in the community. So thank you. Thank you all for participating. We hope you all become a trusted messenger, and we will contact you of how to do this.